Now let's move to the chemical properties, and here comes to the chemical properties, where we have to discuss separately about the reactions based on the carboxyl group and the amino group. Why? Because in the previous slide I have told you that these two groups are acting individually as a separate entities, isn't it? So let's come to reactions based on carboxyl groups. So first we are having the three types of reactions as examples I'm giving here based on the carboxyl group. For example, first one, amino acids form salts when reacting with the bases, and esters with alcohol. That means when amino acids are being reacted with the bases, they will form the corresponding salts. And in the same manner, if the amino acids are going to react with alcohols, they will give the corresponding esters. Then coming to the second property, which is very important in the formation of different biological important amines. One of the most important property of amino acid we can consider decarboxylation, that is removing of the carbon dioxide group from it, is going to be called as decarboxylation. Amino acids undergo decarboxylation to produce corresponding amine. That means in the structure of amino group, in the carboxylic group, if CO2 is being removed, then we will get the corresponding amine, and this amine is made very having a very significant role in the biological metabolism. Okay, so the best examples of this biologically important amines are your histamine, dopamine, adrenaline, serotonin, etc. Like this, we can go on with the different types of examples of amino acids and amines. Sorry, it's amines. This reaction is very significant in the living cells due to the formation of many biological important amines. The third type of the reaction that we are going to discuss is amino acids reacting with the ammonia group. So now ammonia is a compound, and aspartic acid is an amino acid. Here I have given examples of amino acids. When they are going to react with amino group, they will give the corresponding amines. That is, aspartic acid when combined with the ammonia, it gives aspargine. Glutamic acid when combined with the ammonia, glutamine. So that's how these are the few reactions based on the carboxylic group in the amino acid. Then we'll move to the reactions based on amino group in the amino acids. So these are the few examples of those reactions. So first of all, the amino group behave as a base and combine with the acids to form the salts. How in the previous reactions that we have discussed, carboxylic group when combined with the acids they form the salts. In the same manner, the amino group when combined with the acids, it also forms the salts. Example, when HCl is treated with the amino group of amino acid, it forms salts. And then the very important reaction in the amino acids is reaction with Ninhydrin reagent. What is this Ninhydrin reagent? This Ninhydrin reagent is a type of chemical which is especially used to quantify and determine the amino acids and the proteins in the samples. And what is this reaction? So amino acids, when combined with the Ninhydrin reagent, it gives or forms a keto acid and ammonia and ninhydrinodontin compound. So this ninhydrinodontin compound again combines with the ammonia group and one more molecule of ninhydrin and gives rise to a complex called as Riemann complex, as this reaction was put forward by Mr. Riemann. That's why this is called as Riemann complex. And this Riemann complex is going to be of a purple color and or Riemann's complex. And as I told you, this is very important for the quantity and determination of amino acids and proteins in the samples. Then the third type of the color reaction that we are going to discuss is amino acids can be identified as a specific color reactions. Then transamination. What do you mean by transamination? The amino group is going to be shifted or moved from one to the other, and that's how it is changing its structure. Then we call it as transamination. Transfer of amino group from an amino acid to a keto amino acid to form a new amino acid is a very important reaction in amino acid metabolism. Every one of us know about the carbohydrate metabolism because we from the lower classes discuss about glycolysis, TCA cycle, all these things, but many of them don't know about, that we have the other metabolisms also like amino acid, then nucleic acid metabolisms. So this is another type of metabolism, that is amino acid metabolism, that is conversion of sorry conversion of amino acids into amine form or some other form for the metabolic reactions. This is also important. So the transamination is that. Next, coming to the fifth one is oxidative deamination. The amino acids undergo deamination, like how we have discussed in the decarboxylation. Here, the ammonia group is going to be removed, and amino acid or free amino acid. The amino acids undergo deamination to liberate free amino group or free amino acid. So that's all about the properties of amino acids, both chemical properties and the physical properties. And I want to make it note on a special topic called as peptide bonds, which is very important. So here amino acids are linked together by amide groups, also called as peptide groups. In the initial starting of this video, I said the amino acids are the building blocks of proteins. So for the building blocks, there should be some bonding. So that bond is being termed as peptide bond or amide bond. During protein synthesis, the carboxylic group of an amino acid at the end of growing polypeptide chain reacts with the amino group of an incoming amino acid, releasing a water molecule. The resulting bond between the amino acid is a peptide bond. If you observe here, this is one amino acid and this is another amino acid. Now the ammonia group of one amino acid and the carboxylic group of one another amino acid is going to have the participation of the reaction, and this water molecule, that is H2O, is being liberated, and there will be a bridge or the bond formed between these two amino acids called as peptide bond. Here it is going to be called as peptide bond. So a peptide bond is going to possess NHC double bond O. So this is a copolymer of amino acid is going to be polypeptide. Peptide is just the link between the two amino acids. If it is going to be of in a large number, then we call it as polypeptide. Then amino acid polymers of less than 50 amino acids are called as polypeptides, peptides, or oligopeptides, etc. But when we call these amino acids as proteins, means when they are of more than 50 amino acids are being termed as proteins. Remember, this will be very important to understand, because most of the students will have the confusion of what is a polypeptide, what is a peptide, what is a protein. So simple difference between a polypeptide and the protein is, if the amino acids, that is the linkage of the amino acids, are going to be equally or less than 50 amino acids held together, then we call them as polypeptides. If the amino acids or the polymer of above 50 amino acids, then we call them as protein. So that's a simple. So more than 50 amino acids polymer, we call it as protein. Less than or equal to 50 amino, 50 amino acids, we call them as polypeptides or peptides. So that's all about the topic of amino acids. Thank you.